All right, let's do this. It's been almost exactly a year to this day that I submitted my first DNA samples to the lab for sequencing, and here I am now, riding my bike to drop off the biggest batch of samples yet. We started off this journey by venturing to the Ballona Creek Wetlands where the LA River meets Centinela Creek, and I showed you guys how I collected some soil and grass samples to bring to the lab where we cultured them and began the process of creating individual isolates to catalog and study. And now that we're here, back to where Everyman Bio all started, I can't help but to be thankful to have had this opportunity to share it with you all. I'm constantly reminded and humbled by the power of nature to create new pathways forward in our lives, and opportunities for us to integrate our personal experiences into a shared reality welded by the pursuit of truth and science. What discoveries can be had if we can just take a moment to be still, to look, examine, and appreciate the vast mysteries our universe has waiting for us just beyond the horizon. I hope that as my journey to explore the world of fungal genetics unfolds, it inspires you to take action in your own life and pursue meaning beyond the self and into the world around us. Like the kingdom of fungus that grows beneath our feet, you have so much to offer. With a bit of luck, your talents will yield large fruits and provide a harvest to feed your soul for years to come. And I can't wait to be there to see it. Hello friends, we're back in the lab and we're taking a little bit of a break from electroporation to visit our LA River bioprospecting campaign. And I have here all of the isolates that we've been able to successfully isolate from the LA River. If you remember, I went out and collected a piece of grass here, which then we plated and isolated into this fungus. And we also did some soil dilutions and then grew them out on these plates here. So we've done quite a bit of work already in going out into the wild and grabbing some wild fungus and then bringing that back into the lab to create these beautiful set of isolates here. So what I want to do is bring you guys through the process that I have created for managing something like this because, well, it's a lot of information that we're actually creating as part of the DNA barcoding process. Not only do we need to do the work of extracting the DNA out and sending it out to the lab for sequencing, but we also need to collect that data, publish the data, and then we're not going to just publish the raw sequence. We also want to publish pictures and photos of the physical structures of these beautiful specimens as we've isolated them. That's one thing that I've noticed is in the literature, it's often missing color photos. And so a big part of the DNA barcoding process for me is to try to take some decent photos of everything growing on this media and also any microstructures. So we'll take some of the spores and canidia and other structures and look at them under a microscope, take some photos and share those with our data as well. And I share everything on mushroomobserver.org and iNaturalist along with the sequence. But the first step involves assigning IDs to each one of these that we can track through the system that I've created. And I actually have a barcoding Google spreadsheet where I can track each of my specimens and all the metadata associated with them, the lat and longitude, the media that I used, and just all the information that other scientists who might be interested in studying these species would want to have access to. And also anyone who's interested in LA River fungi, because I don't know that anyone has performed a proper survey of fungi in the LA River. Okay, so the first thing is I'm going to basically be organizing this and entering the information into the spreadsheet, and then that will generate an ID that I can then write on the back of these. I'll then be able to take that over to the lab with the flow hood on, open the lids, take some good photos, and then start the process of extracting the DNA, doing PCR on the ITS region of its DNA, and then sending it off to the lab. So let's go ahead and get started with entering this into the spreadsheet, and then we'll take it from there.
Once I have all the isolates, I'll bring them over to my desk and then enter in all of the metadata that I've collected about where I've collected them and enter them into the spreadsheet. And then each sample will get issued an EMB ID that I then write on the back of each plate. And that helps me to just keep track of it from photography all the way to sequencing. Of course, before we do some barcoding, we're going to need to sterilize some of our tools and reagents. Let's review what those are right now. 2 milliliter microcentrifuge tubes, a jar of 1.5 milliliter microcentrifuge tubes, toothpicks, water, 1x TB buffer, LB broth for my electroporation experiments, pipette tips. Now we cook everything at 15 psi for 20 minutes. While the pressure cooker's going, let's take a closer look at some of the video I shot while capturing observation photos.
Now that we have everything sterilized, go ahead and take out everything that we need for barcoding, get it set up over here, and we'll get this thing rocking and rolling. All right, the first thing I'm going to be doing is preparing a 0.5 molar solution of sodium hydroxide, and this is going to be used to lyse the fungal cells. Over here on the right, we have 9 samples that we'll be attempting to barcode today. And so I need to take out 9 tubes for the DNA extraction, and then I set aside another 9 tubes for the PCR reaction. Next thing we need to do is we need to put in 30 microliters of the sodium hydroxide into each of these tubes. Okay, for each one of the samples, what we're going to do is take a pipette tip, and I'm going to light the end of it on fire and stick it here so that we can create ourselves a little pestle for grinding the tissue. And then I'm going to take a piece of tissue and try to aim for the newest growth around the outer edges, and I'm going to put it into one of these tubes containing the sodium hydroxide. One thing that's really important when you're running a lot of samples like this is how you organize them. And so what I'm going to do here is, as I go through each one of these one by one, I'm going to move them down to this bottom row. That'll help me keep track of which ones I've already done. I also have all of these plates already labeled with their ID that I've created in my barcoding tracker. So this is batch 8, specimen 1. And these are already all in order, so I don't have to worry about these getting out of order. And so we'll go ahead and I'll show you how to do that now. finished adding all of the samples to the sodium hydroxide here. Now we need to let them sit for 10 minutes. But I do want to mention that in general, I recommend you do these in smaller batches than I've done here, because it's probably been a good 20 minutes since the first one was first done. So you don't want to let the sodium hydroxide sit and chew at the tissue for too long, or it could potentially damage the DNA. But we'll go ahead and let it sit for a little bit so that these last few have plenty of time to lyse, and then we'll continue to the next step, which is putting in 150 microliters of the Tris HCl or TBE buffer. Now we need to let these incubate at 95°C for 10 minutes. Now we need to spin them down for 5 minutes. Alright, so we've completed the extraction portion of the DNA barcoding process. Now we're ready to start the PCR process. And to do that, I need to put together a vial containing all of the primers and the Taq polymerase that we need. And so I have this little spreadsheet that I use where I can put in the number of samples and a safety buffer, and it will tell me the total volume of each of the reagents that I need for my reactions. So let me do that now. Okay, now what I'm going to do is put 24 microliters of the master mix into each of our PCR reaction tubes, and then we'll add 1 microliter of template DNA from the extraction step into each of these tubes. Okay, we've prepared all our PCR reaction mixes here, and we're ready to start the thermocycler. And we'll check back with you in a few hours when we run the gel. Okay, PCR is complete, and now we can get ready to prepare a running gel for our gel electrophoresis, see which PCR reactions worked. So let's get this set up, and then I'll check with you guys when we run the gel. Alright, we're ready to pour the gel. Now the gel casting tray has 9 combs. We actually have a total of 9 samples, so if I wanted to put a ladder in, I'd actually need 11 lanes if I wanted to have a ladder on each side. So what I'm going to do here is I'm going to go ahead and do a double comb on this. That'll give us 18 wells that we can pour our DNA and ladders into, and I'll just use half and half on each side. So now we just take our melted agarose gel with fluorescent stain in it and pour it into the casting tray and let it cool for about 10-15 minutes. Before we run the gel, I wanted to show you how I lay this out. First thing I did was figured out for the gel what I'm going to put on each row. So start with a ladder and do 1 through 5 on the top row, and then 6 through 9 on the bottom row. The other thing I did was I went and took a look at each of the cultures, and I took a guess on which ones would actually have successful PCR and which ones wouldn't. And that's coming from my experience with knowing that the earlier you catch the culture and the newer the growth is, the easier it is to PCR it. And heavily pigmented isolates as well seem to have some problems. So let's take a look at this run and see how my predictions compare to the actual results. Whew, that was a lot of pipetting. Let's turn it on and let it run.
All right, guys, let's look at the gel and then review our results. Okay, so let's compare the actual results with what I predicted. So, as predicted, sample 2 did not work. 8.4 was unpredicted. That one did not, the PCR reaction failed, but I predicted that it would work. And then 8.8 also did not work. So what do we do from here and what can we learn from this? Well, and also, how did I know that these were not going to work? So let's look at them. So the two that I predicted would not work were these two. What I have noticed is the more pigmented the sample is, and the further along it is in its growth cycle, both of these are producing just tons and tons of spores, but very little mycelium, that the harder it is for the polymerase chain reaction to work. And that could be because they're producing nucleases which cleave the DNA and makes it basically where it doesn't work. The one that I didn't predict did not work was this one, because this one had lots of tissue, it was very easy to pull off, and I think for this one I'll probably just rerun it. But there is a possibility that even this one is gone on too long and is producing something that is inhibiting the PCR reaction, or maybe I just didn't grind it well. But in general, I'm very pleased with this first batch of barcoding. So the next stage here is I'm going to rerun a couple of these. I also have some additional ones from the moth mold culture that I'll run. And I'm going to go ahead and take the ones that did work, put those in the refrigerator, and probably get some stuff shipped off to sequencing in the next couple of days. And this time around, I'm going to use a company called MC Lab, and they want you to arrange your samples like this little diagram shows. So it can get a little confusing because you need to label these tubes A1 through H1 for one row, and then you move on to the next row as you fill those samples out. So I have 14 total samples, so I need 2 strips of 8 PCR tubes. And so I went through and just kind of took the order form and built a draft version of it where I put the name of my sample as it corresponds to each tube. Now what I need to do is I've pulled out these strips, so I have two rows here, and I've labeled them corresponding to this little diagram. And these are all the working samples, and these are also in order that I have them on the order form here. So now I need to go through each one and transfer this sample to this tube, this sample to this tube, this sample to this tube, and just continue on. And then I have this extra tube here because MC Lab wants me to send in primer for the forward primer so they can do forward primer sequencing. So I will put in about 10 microliters of forward primer for each sample, and then I just denote that on the order form. To actually order, I'll actually go to the MC Lab website, I'll log in, I will put in how many samples I wish to order sequencing for, plus PCR cleanup, and then I will submit this form completely filled out, both online, but also when I mail these in to them and they'll receive it, they have a copy of it as well. In the next video, I'll follow up with you on the results of the sequencing, and we'll review what we collected from the LA River and the moth mold cultures. So see you next time. All right, let's check it. Oh wow, that is really cool. I did not expect that.